All right, I've got 1 o'clock, so I'm going to go ahead and get started. My name is Kyle Curtis. I'm an environmental scientist for Hampton Roads Sanitation District, and today I'm going to talk to you all about a part of one of the research programs we have here, which uses microbial source tracking to hopefully identify areas of microbial contamination in waterways. So, get my slide to go here. There we go. So a little bit of context real quick. Hampton Roads Sanitation, if you're not aware, is the regional wastewater authority. And so what we do, we're part of your HROBS bill. If you're living in Hampton Roads or the Middle Peninsula or anywhere in Southeast Virginia, we're likely treating your wastewater. Between the 9 major and over 7 or 8 sort of smaller scale facilities, we have the combined capacity to treat like 250 million gallons of wastewater per day. And so we service the whole southeast part of Virginia. I work in the water quality department, so I don't have a hand in treatment plant operation. What my job primarily is to be part of some of these water quality research initiatives that HRSD does. And so that's where this kind of research sits in terms of HRSD. It's in the water quality department. And that's sort of who's giving you this talk today. And so the context for all of this before we get into source tracking is just microbial water quality in general. And so what I've got up here is sort of a cartoon depiction of the landscape with a lot of familiar features that I think, we would recognize, right? Being down here in, if you're in Norfolk or Virginia Beach, there's water all over the place. The ocean and the bay are right there, but then we also have large estuaries kind of almost on all sides. And so when we think about microbial water quality, one of the things we want to think about is if we're going to deal with or be interested in the ways that microbes might contaminate water. And so when I talk about contamination of water via microbes, we're thinking of, bacterial pathogens, viruses, protozoans, things like that can get you sick or can have a negative impact on aquatic health. And so as we start to think about that, some of the inputs might be obvious and some maybe are less obvious. And so kind of right off the bat here, you can see we have the wastewater treatment plant, which we know treats water before it goes out into the environment. But then we have all of these other sort of ways that microbes might make their way into a water body and serve as a source of contamination. And so one of the big ways just generically is runoff from rain events. So stormwater accumulating on land, running across the land, picking up contaminants and discharging them into waterways. And so kind of on one side of the equation, there's, runoff of microbes of animal origin. So livestock runoff that includes the feces of like wild animals. So for instance, deer or foxes or anything, it kind of depends where you are, right? What animals you have, but any variety of wild animals, even in a suburban environment where you have birds, raccoons, possums. So there's all of that, right? That can run off and make its way into whatever the nearest water body is, whether it's a river or the estuary, or, if you happen to live right by the ocean. And then on the other side, there's human contamination, human source contamination. So now we're thinking about sewage, right? So here we'd be thinking of either a septic system or, sewerage infrastructure that is leaking for some reason, because it's 80 or 100 years old, or maybe there actually has been a pipe failure because of something, right? There was drilling that hit a pipe, whatever it might have been. Or maybe we had enough rain that the treatment plant could not actually receive all of the wastewater. And so some of it got bypassed and did not make its way in to the actual treatment facility, meaning some of the sewage actually just hit the waterway untreated. So there's a variety of ways that both animal and sort of human origin microbes can make their way into waters, right? And any of those are of concern. The other part of this is you can see all of these brown arrows on this slide. Once that microbial contamination has made its way into the water, there's all types of transport mechanisms that can either enhance or discourage, your ability to quantify microbes, whether you'll see them or not. and where they get transported. Are there any downstream impacts? Everything from tides to ocean currents to wind moving water around to even, sedimentation and resuspension of like sediment associated pathogens in shallower water bodies. And so, like a lot of the problems, this is a science and engineering talk. So like a lot of the problems that you likely deal with, this one also is complex and has a lot of pieces. And so we're going to kind of break off one particular part of the problem and focus on that. But I just wanted to give a background for this whole idea of microbial water quality, because that's where sort of all of this rests. And so I just spent a couple minutes talking about the ways that microbial pathogens might enter waters. And so our response as a community, this is based on guidance from EPA, is that we're going to monitor all of these water bodies, right, depending on their intended use. And so the idea here is the level of contact that you'll have with the water is what should drive how stringent the requirements are for it. So to take the obvious case, with water that you're going to drink, we want the most stringent requirements, and that's what exists, right? There is no tolerable level of pathogens in water that comes out of, that's coming out of your tap, right? But for water that you have less contact with, so maybe, it's a recreational water, something like an ocean, like the ocean, for instance, that you could take your kayak out on or go surfing or whatever. The monitoring requirements and the water quality criteria are then based on that intended use, and this you can kind of go on down the line with this. Something like a stormwater ditch behind your apartment building, that's not a recreational water body, right? The assumption is not that you're gonna take your son out there and like go for a swim. And so overall, we monitor microbial water quality to protect public health, right? We don't want anyone to get a waterborne pathogen, basically. And if if you're from around here or anywhere, near the water, you've probably seen on the news or you've seen in person the signs up that say, hey, this beach is temporarily closed due to high bacteria counts. That is this program in action. Like that's this happening. There's routine sampling, and then these tests, which I'm getting ready to describe, get run. And if levels are above what has been, studies have shown to be sort of indicative of of a high likelihood that you could become sick from swimming in it, then that gets closed, right? And so the way that we do this, we have done this for a while, and we continue to do this monitoring of microbial water quality for all these different, sources of water, are to use these culture-based tests for bacteria. And so what culture means, if you're not from the micro kind of world, is you're actually going to grow up viable bacteria and count the colonies. And so you'll use E. coli for freshwater, and then you'll use, a set of enterococci species for brackish or marine waters. And the only difference there is that they're a little more hardy to a variety of salinity. And so you want, you want something that survives in the water so that you can have a test that actually tells you if there was some kind of contamination. And so the idea with these indicator bacteria are that, yeah, there are pathogenic strains of E. coli and enterococci, like, of course, but we know those aren't the only thing that gets you sick. So why do we only measure those? And the answer is there's just too many potential viral pathogens or bacterial pathogens, or like I mentioned, protozoa. You could get cryptosporidium or something. And I'm not saying all of these things are all in your water all the time. Obviously they're not. We have really good water quality around here. What I'm saying is you couldn't run the suite of tests to measure directly all of those pathogens. And so what you need is something that you can measure by itself that can give you some idea, that's indicative of the other ones being present. And what we came up with, some years ago was these kind of sets of fecal bacteria that are present in your gut. And so the idea is, and the studies show the association is there, that if E. coli is present in the water, if enterococci species are present in the water, especially at like a high concentration, then we should start to worry, are these other gut microbes there, right? Do we have reason to believe that, yeah, maybe there's some, microbial contamination? And so what you see here is just a simple picture, this is out of a Nature or a Science paper. This guy doing a grab water sample. This is just what it looks like out at Virginia Beach 2, although, it's probably a little warm for all that gear, but the dipper pole and sampling technique would be very similar. And this is a plate where after you filtered your water sample, you would put it on a Petri dish type thing with a media, and you'd incubate it, and you would count those colonies. And so, like I mentioned a minute ago, when we talk about a culture-based test, that's what it is. You're growing up live or viable bacteria and counting them. And so that's what's done. And that has a lot of benefits. There's a lot of benefits to that. One is the one I described where, you can measure for one thing and get an idea about all the others maybe being there. The other is that, it's really not super complicated in terms of lab technique, right? I mean, you need to learn how to do it, but it's not operating a very complicated machine. It's not a mass spec or something crazy like that. And it's not expensive either. That the media to grow these things up on and the filtration setup is not that expensive. And so for all those reasons, knowing that we need to monitor a ton of water all over the country, the fecal indicator bacteria, do a good job. Like, they're a good solution. But we also know, especially as more sophisticated techniques have come along, is that there's some real shortcomings to using fecal indicator bacteria to indicate the presence of, recent contamination. So one of the problems is that these indicators, E. coli and enterococci, are ubiquitous in the environment. And so what that means is that they're just out there anyway. If you go out and sample and see, oh yeah, we actually got a hit for enterococci, that doesn't necessarily mean that recently there was some kind of contamination input. And that's because these things can grow outside of your gut and just persist and even multiply in the environment, particularly in the areas that we likely would sample, unfortunately. And so that's a limitation, right? Because you want to test that when you run it, it only tells you about the thing you're interested in. It only tells you about the recent contamination. It doesn't tell you about what the background level is. But that unfortunately is a shortcoming of these indicator organisms. The other thing, I mentioned that they can persist and even grow in the environment, is that these tests, the E. coli or enterococci, is not host specific. And so what that means is both of these, E. coli species and the enterococci genus, you have these, these microbes in your gut. They're commensal in your gut. Not all strains, but you have some. But so do a lot of warm-blooded organisms, right? So like all of the wildlife that I mentioned at the beginning of the talk, your pets, livestock, all of it. And so any runoff that would, mobilize fecal contamination of another sort would still show up when you ran your fecal indicator bacteria test. And so basically the gist of this is, if you go out and run that test, that membrane filtration culture-based test, you'll get a concentration, right? You'll get number of colonies per 100 ml of water, but you'll have no information about where that bacteria has come from. And there's some real benefits to being able to do that, which we're going to talk about. The other side of this, and the tool that we use that I want to focus on, is this HF183 molecular marker. And so this is different than the fecal indicator bacteria in a lot of ways. So the big one is that it's human specific, right? If you run this test and it shows up positive, you actually have a real good indication that that water that you've sampled has some sort of sewage contamination, right? You don't have to worry if that, maybe that's dog or something, maybe this is runoff from, I don't know, a nearby dog park or something like that. The other thing is that it's DNA based. And so while that makes it a more complicated test for sure, it also means that you won't find this signal persisting in the environment for like weeks or months. Like, it's certainly not going to multiply. If you know anything about nucleic acid, it's not super stable. And so what that means is that if it hits, a water body, so say it hits the estuary, for some reason, it makes its way in there via whatever, a leaky pipe. it's not going to be there for very long. There's a bunch of studies about this, and it can certainly vary, but I mean, the estimate I've seen is on the order of like 96 hours or something. You know, we talk about it on the order of hours, not weeks or months or longer. There's no naturalized colony of this because this is DNA, right? This is, you're not culturing a bacteria. And so the point of all that is it's more specific, not just in the fact that it targets, you know, just human source kind of sewage, but it's also specific in the sense that if you detect it, it's probably a pretty recent or a pretty steady input. And those two things actually really make this a very useful tool for identifying infrastructure issues, which is kind of where all this is going. The other part of this is that there are qualitative versions. So there's a presence absence version of this test. And when it first got created, that's kind of all you had, which even that is pretty good. But when I mentioned some of the case studies, you'll see why it's really valuable to have a quantitative test. And this is quantitative. You do get this in terms of concentration. So in case you're interested, the unit is, you know, it's copies of the target. So you'd say copies per 100 mil or something like that. And so all of the case studies that are the second half of this talk are using this HF183 marker, this molecular marker, which I should have mentioned uses either qPCR or digital PCR to quantify these DNA targets, which only would have gotten into the environment if there was the presence of sewage of some sort, some human-specific contamination. And so to really just kind of drive this point home, O'Mullen et al. Put out this paper that I thought was really useful for summarizing what the predicament that all of us that want or need to monitor water quality are in, which is that you can have autochthonous bacteria, which is, you know, just a very long word for saying it's already inherent in the system. So that's like the naturalized strains that are just there. And, you know, your test will measure those, right? And then down on the bottom half of this figure, the second and third sort of rectangle there, allochthonous, so like outside of the system that are getting put in are runoff from your agriculture or your animal sources, right? So your test will measure those. And then there's human sources and your test will measure those. And if you run just the basic fecal indicator bacteria culture-based test, again, you'll get a concentration, but you won't know which of these three panels or what combination of these three panels you're working with. And so that kind of leaves you with two sort of problems. So one is that ideally, if you find a high concentration of this bacteria, which is suggestive of, you know, a public health concern, you should want to direct some sort of remediation efforts, right? So you should want to try to correct the problem. And so if you don't know what the source is, the remediation efforts are really tough because they differ greatly. So if you could know that this is primarily dog waste, then your remediation effort is like public outreach, you know, scoop the poop campaign, right? Like you might have heard these things, or putting in waste like a dog pickup sort of stations at the dog park just to get people, make it easier on people or whatever it might be, versus figuring out that this is like largely a human input. This is largely sewage. Well, you know, don't waste any money on the sort of scoop the poop campaign. Go out and figure out like what's going on with your sewage infrastructure, right? And so that's one of the problems with having a largely non-specific test. And then the other piece of it is that a lot of pathogens, like these pathogens that we talk about, whether it's a bacteria or a virus or whatever it might be, not all of them, but a lot of them are host specific. And so that's important because if you could only, you know, if you wanted to start prioritizing what sources of contamination should we go after first, you should want to go after the human sources first, because human source contamination, just because, again, the pathogens, you know, a lot of them are host specific, is way more likely to get you sick than, you know, bacterial contamination from other sources. Now, I'm not saying, you know, you want to swim right downstream of like a big ranch where they have cattle, not what I'm saying. But if you were to really prioritize, you know, human source contamination is definitely what does drive the public health risk of overall microbially contaminated waters. And so this is just kind of a summary of that beginning part, which is that microbial source tracking, which HRSD is not by any stretch the only group doing this. This is sort of an established term in the field of environmental microbiology. And this is just one summary, right? It's just one possible way to think about it. It's a set of tools to discriminate sources of fecal contamination in the environment. So HRSD has a molecular lab. We're able to quantify a huge long list of bacteria and viruses and all kinds of pathogens in all these different matrices. You know, we have really good molecular biologists. We can design our own assays. And so what we thought is that we should employ these techniques out in the world to try to see, hey, if someone who is already monitoring a water body is seeing like chronic high bacteria levels, but they only have the culture-based test, they only have the really basic version that can't tell them, you know, is this recent, is it, you know, and what the origin is, we should get involved and use these tests to try to see if we can help figure that out. And so just to point out that EPA actually does have an approved method for this HF183 assay. And so for anyone who's super interested, you can find that. It came out in 2014. So this was in the world of research for a long time, but it is so effective and useful and used so widely, at least among researchers, that EPA actually did put out a document giving a recommended way to not only use the approved set of PCR primers and probes, but analyze the data. So that is out there. It is actually in the world of regulation. And so now probably the more fun part, which is how does this work out in the real world? And so up here in the top left, you can see this graphic from Reicher et al. From 2011, which should tell you how long this stuff's been around. That's definitely not the first paper to come out about this, and that was already 10 years ago. But what they're pointing out is that, okay, you should start by thinking about your pollution source, which is kind of what I've been talking about for 15 or 20 minutes. Do you think it's kind of like wildlife? Do you think it's agriculture and confined and it's these certain species, or do you think it actually is human? Because again, the way you proceed probably hinges on that to some extent. And then what you might do after that is, you know, in this area, not only because we don't have a lot of reason to think there's a lot of wildlife inputs. I mean, there are certainly some, and there's not a lot of agriculture, at least down right where we are. This is, of course, not the case out. You don't have to go too far west for that to be different. But also because we know that, like I mentioned, human source contamination drives the public health risk. We want to start with, let's identify that or rule that out if we can. And so what we would want to do is, you know, if a group, like I mentioned before, was monitoring a water body, we would want to go out to that water body, and then we would want to start sampling upstream in the stormwater infrastructure. And that's what this picture is, the second picture right here. I'm not sure if you can see my cursor, but that dipper pole with it's got a bottle on the end of it, that's sampling in the stormwater infrastructure. Because what we learned is that even though we would likely be looking for sewage that has come out of sewage infrastructure, right? The fastest conduit that water typically finds once it's sort of loose is something like a stormwater pipe. And so if we have a line crack or worse, it typically finds its way to the stormwater pipe. And so by sampling the stormwater, we kind of reduce the issue of getting our signal diluted out in a big water body, and we can maybe run into where the problem is. And the other good thing is, like, we have really extensive GIS mapping of the stormwater and the sewage infrastructure. And so we'd go out and sample kind of upstream of the area, and we would run our molecular tests, so HF183 at least, and maybe some others. We would also quantify, we would do some general tests similar to the fecal indicator bacteria so that we have numbers for both the human specific and the general test. And then we would take a look at our GIS and say, does this make sense, right? Is our place with the highest number right by where some sewage infrastructure crosses? And if so, maybe we need to investigate that and see if that is, you know, an obvious problem. And so that's what we do, you know, to boil all of that down to just a couple simple points. What we would do is take the latest molecular assays, so the best version of that HF183 assay, which, you know, we can run and really have a lot of experience running and are really confident in. We take what we know about the collection system because, you know, at the end of the day, we are a wastewater utility. And so we have a lot of in-house knowledge about how all that stuff works. We have great mapping of it and access. And we kind of integrate those two things to do some adaptive kind of in-pipe targeted sampling and investigation. And so for the other part of this talk, I want to just show a couple kind of examples of how this works in practice. We've had a ton of these studies with lots of successes, and they're always ongoing, but here's a couple that kind of illustrate how it works. So this was actually the first one, and by chance it happened to be one of the really clean examples of how this thing can go. So one of the local, I think, nonprofit or environmental groups had been sampling up in this area. If you can recognize the Lafayette River here, this is Norfolk, terminals, Hampton Boulevard Bridge. Up here is a tributary of the Lafayette called Wayne Creek. And so during some sampling up here, they saw some really high bacteria values. So not just ones that are, you know, maybe there's a little bit of a, you know, stormwater runoff or right after wet weather. Like, no, these were like real high numbers. So we said, okay, we're going to take our molecular tool and we're going to figure out how to apply it here and do some source tracking. And so the first thing is this is that same location. This is the headwater of the creek, but now what you see is the upstream stormwater infrastructure in blue. So, I mean, it's probably no more complicated than any other part of the system. Norfolk's pretty flat, so you try to get the water to run off any way you can. But what we did is said, let's see if there's any upstream inputs that we can identify using HF183 to figure out why at the receiving water or the discharge point, whichever way you want to think about it, we're seeing these really high bacteria values. You know, if we never find anything, maybe it really is some other kind of general fecal input and not a human source. But we thought, let's just see if we can find out if it is human source. And so we did all kinds of sampling because this, like I said, this is the first time we got out there and did it. Got down in the pipes to figure out where they actually went, because sometimes, you know, the GIS layers just aren't perfect. I think that's Raul crawling into a pipe. We install equipment because sometimes we want to do flow pacing and see what the load is over the course of time, not just do a grab sample, or we might want to collect samples from an auto sampler, you know, over the course of days. So we did all kinds of stuff as we were learning how to sort of implement this in-pipe microbial source tracking out here in the real world. And so here's a crazy looking picture from exactly the same area where I've just turned on the sewage infrastructure, and that's in this red and yellow kind of dashed line. And yeah, don't squint too hard at this. I just wanted to point out that the sewer gravity lines in the area, which the red and yellow, are obviously all over the place because this is a residential area and you need to move wastewater to the nearest pump station and then out of the area. This green line is an interceptor line. This is where HRSD picks up wastewater from the locality sewer and moves it towards the nearest plant. But I just put that up there to show that there's lots of opportunity for, if the origin of this high bacteria value downstream was from a sewer pipe, there's plenty of sewer pipe. And so we need to really be methodical about how we try to rule in and rule out areas of interest. And so what we have here is a really, really stripped down version to kind of make the point. So we would start downstream and work upstream, you know, downstream where we actually saw our high bacteria value, and work upstream at every intersection, trying to either rule out a leg or follow the signal as best we can. And in this way, kind of narrow down the search area before we start, you know, trying to figure out, all right, what is the actual origin of this? Because like you saw on the previous slide, there's too many pipes to look at each of them and test all of them. And so if you see poor water quality up to there, then you're going to sample both of these branches. You're going to get results and find out that one in green, that's a non-detect for HF183, but the one that's kind of straight in line in red there, yeah, that's still really hot, still a high concentration there. So let's continue until we can rule out everything and until we stop seeing the signal, basically. And this is a simplified version of it, but this is exactly how the process goes. We go to each intersection, sample all the possible tributaries, the upstream pipes, and just keep tracing the signal until we no longer see it, and kind of box in the area that we think is worth further investigation. So here's some real data from this area. So this is actually from July 2014. So this is a while ago. But what you can see, this area down here in red, kind of by the headwater of the creek, just where that star was on the other maps, we saw with the HF183 marker, everything in this main sort of stormwater pipe, or sorry, stormwater ditch was non-detect until we got to here, and everything on this side was non-detect. But kind of moving north, we got detections. And so just like sort of on the previous simplified schematic, we move upstream. And this is from September, but we didn't wait until September to investigate this. There's many iterations of this sampling, but once we got to September, we had kind of traced the signal to its extent. So at this point, we've moved upstream and we ruled out this side, this east kind of branch. We got up to this junction. We ruled out this one that goes kind of north and west. We ruled out this set over here on the east, and then we stopped seeing it. Up here north of this sampling site, we did not see it any longer. And so now we're thinking, okay, the problem is somewhere in here, right? Because like I mentioned, there's not a background HF183 signal. So if you're seeing it, and you're seeing it months later as you conduct this sort of methodical rounds of sampling, then there's something going on there. And so same map down here. Now we got to figure out, all right, let's do like another level of analysis. Now let's throw the concentrations on there from all of these individual samples and see where on this run of pipe, this connection of pipes, might the source of this be. And so this is where looking at the concentration, the fact that this marker is quantitative is actually really useful, because just looking up here, you might think, okay, well, there's the source, right? You don't see anything above it, but you do see stuff downstream. But if you look at the concentrations, what you actually find is somewhere down here, somewhere in that area is the likely source. These are all concentration and copies per 100 mil of that target. And so we're getting 1 million copies down here. It kind of reduces almost by a log each time you get to a junction going that way, and reduces by 2 logs as you go that way. And so what we kind of learned is that, okay, well, the source is actually down here below where we thought it might have been. But we probably have a tidal influence of some of this area causing whatever is going on here to actually get pushed upstream occasionally. Because we know Wayne Creek is, we call all these rivers, but it's really an estuary. And so we have a tide change there. And so now we have a much, much smaller area to look at. We can actually reasonably do things like smoke test the pipe, or have the city smoke test the pipe, or inject dye, or do some way to find out, hey, do we actually have a problem with a sewage pipe in there? And turns out we did, right? This is a gravity line that was near a pump station. We just kind of got boots on the ground, and we're eventually, because we were able to constrain the whole area, we were looking to a smaller region, able to get out and kind of see gray, blackish water bubbling up and find it. And so the city is really happy that we could find this, because, then they can go out and fix it. I'm not saying it's an easy fix, but finding this specific area to go out and replace really does a lot to fix the problem. And we confirmed that with rounds of sampling after the repair was made. When I think we did like 3 or 4, it's been a while, but I think we did like 3 or 4 rounds of sampling just to make sure. And what you can see is once the repair is made, once the pipe's now intact, everything's below detection. And so this is a really good, positive sort of first real world application of this process for us. We go through one other one in Norfolk here that was a little bit different, and show what the difference in, getting a few years of experience under your belt looks like. So we do these all over. This just happens to be another one in Norfolk because I just had the information kind of readily available, but we do them all over the region. If you recognize Colonial Place, ODU is just over here. Here's a Grammy Street Bridge in Willowwood. This boat launch is called Haven Creek, And similar kind of situation with this one, where there's routine monitoring by, I'm not sure if it's volunteers, like citizen science groups, or maybe it was ODU, not 100% sure. But we got tipped off like, hey, can you run some, some of your human marker in this area and just see, same question, do we have any reason to think that it actually is human source or is something else going on? And so what we did is go upstream in that area and take a look at all the stormwater inputs. And so this is just that same map, just pointing out kind of where we are. Now zoomed in, stormwater infrastructure in blue. So hard to tell because each of these little boxes is a junction box, even if no other pipes kick into it. But a lot of this kind of main trunk, as you might expect, just spits out under the boat launch in a pretty good sized pipe there. We had some small inputs up here that we had to check out, but this is kind of our main discharge. So we're kind of working our way up from that. And this is from 2018, also a couple years ago. Here's what we got. So same area, here's the boat launch right here, here's Haven Creek. And, now we've got dots on the map representing sample points from this particular day in April where, green hopefully intuitively indicates a low concentration and up to dark, scary red indicates higher concentration of the marker. So yeah, first of all, at the main outfall, we do see a high concentration of HF183. And so not to beat a dead horse, but if it's there, we know there's something going on. And then if it's there at a pretty high concentration, what that kind of tells us and what we learned over time is that that's either, active and or we're pretty close to it. So the idea that this marker doesn't persist very long means that the different concentrations sometimes can give you an indication of spatially how close you are to it, because it does degrade somewhat as it travels, but also just the effect of dilution, right? The effect of other water that doesn't have this signal in it should dilute it out some. So all I had to say, if you see a very high number, and just for reference, HF183 in raw sewage, so like headworks of the treatment plant totally untreated, is around like 1 million, it's like 10 to the 6, something like that, give or take. If you see anything like that, you're like right over top of a real problem. And so that's like a benchmark that we use when we think about like, hey, we saw this at 10 to the 5th in this pipe. That means raw sewage hit it and it's only been diluted by a factor of like 1 log here. So we know we're like close to a problem. So that's just kind of a frame of reference for how we use the concentrations that also help dial us in and give us pieces of information to track down the source. So blue lines on the map, still stormwater, yellow and red checked are the locality gravity sewer lines. And then HRSD has these either gravity or force interceptors. So again, that just means that's where HRSD's pipe starts. Force main means it's actually pumped. It's under pressure. Gravity means it flows by gravity. So no pressure there, just flows by the slope of the pipe. But you have a lot of sewer infrastructure in this area. So high concentration at the outfall, higher concentration upstream, and then concentrations here that seem to, more or less reduce as you move that direction. And so again, just like before, we're in a pretty low-lying area, and we know that this is a tidal water body. So if our issue is here, it's not unreasonable that we would see both evidence of it downstream, but also evidence of it upstream, because as the tide would push up, you could see the effect of some major problem here, which a lot of zeros on this, but this is a really high concentration result. And so what we found was, in this case, not a cracked sewer pipe. So this is actually stormwater infrastructure. So this is one of those curb drop inlets, sort of rectangular boxes in the curb line that you've seen. You can kind of pop the lid, and this water being this gray, and I know it smelled bad because I was out there that day, indicates sewage. And so we're thinking, I wonder if it's somewhere upstream. But then we're thinking, what is this pipe? This isn't a common thing to have just a, like a 2 or 3 inch PVC pipe right with discharging right into the stormwater like this. I mean, it's not unheard of, and it doesn't immediately tell you that it's something illicit, but when we sample directly out of there, so put our bottles straight under this, no dilution coming right out of the pipe, we find that actually that is an illicit connection. If you sample it enough times, you find out that it's wastewater, and the facility may not have known that they had this illicit connection where somebody had plumbed something from their facility straight into the stormwater rather than plumbing it into the sewage infrastructure like they should have. But that's what it was. And so that's pretty gross, but it is also something that the city can get right out and fix. And that's what happened. And we saw things clear up. I don't have the post sampling on here, but we saw that problem also clear up kind of straight away after that. And as you can imagine, it's pretty satisfying, not only just from a sort of seeing the process work, but also being someone who kayaks right in this water, it's nice to know there's one less input there. And then have one more case study here and then wrap up, and then time for as many questions as are, we have minutes left if people have them. So those were both really small scale examples, because I think this works really well at a small scale when you can get up and sample all of these branches of the stormwater network near to a known problem, narrow down your area, and then start doing some like intensive investigation. So putting smoke in the sewer and seeing if it makes it out, putting dye in and see if it makes it into the stormwater. But something else that we tried was, what if we took the watershed of a pretty big river like the Nansemond River? So this is Suffolk. Well, part of this is Suffolk. This is the Nansemond River coming down here. And said, we know that there's been closures to the shellfish beds that are pretty chronic. They come up all the time, or they've just been closed for a long time and have not been able to be open. What if we did like a big scale surveillance of this watershed and just hit all of the inputs that we knew, where stormwater dumped straight into the river, at least as many of the big ones as we can? Could we then identify some bigger problems and then go kind of apply the source tracking methodology there? And so that's what this is. We went out, sampled a lot of sites a couple of times. I think we did each of these 3 times, and just said, could we identify, in this giant watershed, any of these big problems that we could then like narrow down on and try to figure out what the real smoking gun is, which we always use that term, but it's rarely that. I showed 2 examples of that because they're easy, but some of these things become a lot more complicated. And so I shouldn't have flipped. What we found was lots of sampling up here and up here. I didn't turn on the sewer storm infrastructure or the sewer infrastructure because it's just too crazy, but up in this area away from sort of downtown Suffolk, we didn't see any problems. We saw these sort of fleeting signals up here that we investigated, and nothing that we could find came of it. So a little bit of a mystery, but also not a big scale problem. But down here, we saw a number of pipes with high concentrations, suggesting that there's probably a few source tracking projects to be done down in that area. And that's what we did. We've done that over years and corrected or helped the city identify so that they could fix numerous problems. And so what that might look like if you zoom into the downtown area is, so there's 58 kind of coming in and turns into Constance Road here. Here's a couple of those large red dots. I think the legend is cut off now, but basically high concentration areas that each of these we can go in and say, maybe this is its own source tracking project. Let's go upstream here and see if we can identify the problem. And let's do the same thing over here. And this has also been pretty successful. We're really happy about, the way that worked, that approach of, wow, the watershed's way too big to apply the really intensive sampling to every pipe. But if we just do a couple rounds of that, can we identify the most important areas and then zoom in on those? And that's what we're able to have done and still do. And so, yeah, this is just more examples of the same. We identify sort of the hot kind of segment of the pipe and continue to zoom in until we can land on, the source of the problem, hopefully. So our framework, includes this. We've had a lot of successful case studies, and over the years, and you can see that first one was 2014. So we're over 7 years old for this, and it's been going continuously. We've learned that if we can reduce the effects of dilution, we really increase our chances of seeing that signal. And so I know I mentioned that a couple of times, but that's why we're in the stormwater pipes versus open water bodies. And that's why if we see a faint signal, we're going to go upstream and see if we can get closer to the source of it, because we want to reduce dilution and help us zero in on what might be there. Inhibition refers to inhibition of the PCR reaction. So there are certain things that show up in these matrices. So in estuarine water, stormwater, and definitely wastewater that inhibit the amplification of the target. And so there's ways that we can try to avoid that basically keep us from masking what is a real signal. That was kind of something we had to learn. An adaptive approach was something we kind of always did, which was that rather than, like a more classic kind of study might just pick sites and stick with them, we always said, let's follow the signal until we no longer see it. And I think that's been a really effective way to kind of keep pivoting until we narrow down the area that's important. In-house data turnaround is just a thing that we're fortunate to have. We don't have to send out our samples and wait for turnaround or interpretation. And if we see something weird in our data, if something strange shows up in the QC, we can interrogate it right in-house and figure out what's going on. And that's a huge benefit that we're lucky to have. We also use some kind of simple, this is engineering tools, but really just sort of approaches for saying, if we put a flow meter in, can we get an idea of load? And then we work our way back to some kind of estimate of what percent sewage must have come by this site to give us this concentration. All of that amounts to a C1V1 calculation. So it's not really that complicated if you do put metering out on the pipe and get flow, and you can also get concentration. And then pooled resources, this just refers to, we read a lot of the literature, we reach out to other researchers, but we also learn a lot from our local utility folks and the city utility folks. And that's both of those last two bullets, pooled resources and understanding how infrastructure fails. We learned a lot about what causes these pipes to fail and how you could notice it based on, say, wet weather events that cause a pipe to leak only a certain amount of time because the pipe has to get totally full and press sewage out the top versus just leaking out of a crack in the bottom, for instance. And so in doing this for 7 years, we definitely picked up some successes, and we definitely picked up some skills in getting better at doing this stuff. And so that's why I wanted to bring it to this group, because I thought as scientists and engineers, this is a really, I mean, at least to me, it's both satisfying in the sense that you can see a real application of your work right away if your work helps fix one of these things, but also it's just a cool intersection, I think, of science. So, the molecular tools with engineering, which is all the wastewater and stormwater engineering that goes into this infrastructure and kind of understanding it better. We have 2 publications out about this so far. The one on the left is a real good summation of what I, hopefully just described. And the one on the right that was in Environmental Science and Technology was something me and Raul came up with for kind of assigning like a confidence or a reliability to a source tracking result. And so if you're interested in that, I'm happy to chat with you offline. There's definitely not enough time to go over it now, but these publications are out there if you're interested in them. And yeah, I'll go ahead and wrap up there. I think we've still got a few minutes, but I'm happy to take any questions. I'm not sure if I need to check the chat or what the way to go is here.
If there are questions, they'll pop up in the Q&A for you.
Gotcha. Okay, so no questions right now. Cool. You guys gonna let me off the hook? Okay, there we go. Oh yeah, so a question from Dr. Dewara about, we're talking about stormwater here. I talked about stormwater for a lot of this hour. Any plans to understand and avoid or remedy what happened this week in Germany? Yeah, that is way beyond what I could comment on. Not to dodge the question. Yeah, there's a lot of talk in this area that I've been privy to, and I'm not even an engineer, about how to deal with just the fact that we have so much water and where can it all go when it comes in, whether it's oh, so Tyler Kaysen would like to answer this question live. Yeah. If Tyler's able to do that, I'd say go for it.
Oh, no, I was just clearing it from the queue.
Oh, okay. Gotcha. Sorry. Yeah, so rather than rambling about this, I think the short answer is I don't think sort of stormwater conveyance is something that HRSD has a hand in, other than understanding it better as it affects our ability to treat wastewater at our facilities. And so even though stormwater and wastewater are separate in this area, stormwater does still make it into the wastewater pipes and cause an issue at our treatment facilities. And so from HRSD's perspective, even though we do need to deal with how to handle all the runoff and the pooling water that sort of inundates the area when we get a storm, I think our focus right now is just how to handle those really big increased flows so that we don't have backups upstream or have to bypass. And that's the more we have unusual storms, I guess I should say, the more big storms become typical, the bigger challenge that is for us, but it's on our radar for sure. I'll move this. Let's see. Oh, sure. Yeah, so the other question was, can you talk about how COVID-19 is found in the water system? And I intentionally did not give this talk, even though this is what Raul and I and our team have been up to really since March of last year, because I thought that he might be doing it. And so I just picked something else. But very briefly, we've been monitoring for SARS-CoV-2 in wastewater for this area since the second week, really since like March 11th of 2020. And the short version of this is that very early on, there was clinical data to show that this virus, even though it's primarily a respiratory virus, is shed in your feces. And so once we knew that, we knew, okay, we'll be able to detect this at our plants, because we have the ability to quantify even an RNA virus at our plants. And so we just need to get the correct assays and optimize them. And the folks that work in that lab were like unbelievable. I think they had it done, in an afternoon. And so from there, it's just how much can we monitor? How useful can it be? And so we've been trying to do that, quantify changes in viral load to our facilities, share that information with the health department, because with just the last minute, I want to kind of mention that screening for SARS-CoV-2 in wastewater is super useful, because you'll detect both asymptomatic and symptomatic folks. And so it doesn't replace the clinical testing. But for people who are asymptomatic and never go get tested, you never find out that those people actually had the virus. Whereas if you quantify it in wastewater, it doesn't matter if you're symptomatic or not, you'll shed it. And so when we track trends in our regional wastewater, we kind of have all of that. We have like sort of the community scale, basically, idea of what's going on. And yeah, we've been doing a lot of that. So I'm happy to also chat about that. My email's here. I don't want to cover it up, but anyone who's interested, feel free, or in what I talked about then, or the wastewater work, or SARS-CoV-2 work, feel free to shoot me an email. Well, if we don't have any other questions, should I go ahead and stop sharing, or want to let it go for another minute or two?
Nope, you're good, my friend. I appreciate it. Thanks so much.
Yeah, thanks for the time. I hope everybody else's session goes well, and I'll catch y'all later.
You got it. Thank you.